Hello, everyone. I will be discussing marijuana concentrates and their prevalence and use in today's society. Concentrated forms of cannabis have been around for thousands of years, with reports of hashish use found as early as 10,000 BC in Taiwan. Hashish is a preparation of the stalked resin glands or trichomes from the marijuana plant, and its use spread from South Asia through China to Morocco, where the majority of it is currently produced and exported. Newer methods of extraction have allowed for proliferation in recent years of stronger concentrates like hash oil. Hash oil arose in the 1970s with a THC content up to about 30%. Current medical grade butane hash oil is available at THC content above 90%, with regulatory measures only recently catching up to its production and use. News surrounding marijuana concentrates has not been entirely positive, with reports from Los Angeles and Detroit identifying individuals who suffered from psychotic episodes following marijuana wax use. When interviewed in the hospital, 17-year-old Josh was quoted as saying, "I felt like I was going to die. I kept seeing lights." Reports from Stanford University and UCLA identified additional cases of marijuana wax-induced psychosis. Patients had to be treated with antipsychotics like risperidone until psychotic symptoms vanished. Cessation of marijuana use afterward coincided with no further mental health issues in these patients. Higher concentration of THC in newer marijuana extracts has led to recent developments in the legal situation surrounding these substances. The use of butane as a concentrating solvent began around the year 2000 and was originally protected by the marijuana processing statute here in California. However, a 2008 court case, the People v. Bergen, indicted cannabis blasting as operating a drug lab. Butane hash oil remains sold in California dispensaries and is legally produced in states like Colorado under lawful regulation. Most recently, the DEA classified marijuana extracts as a separate Schedule I drug in order to better track its use against flower marijuana. This classification included both THC and cannabidiol extracts, some of which are medically available in other parts of the world. In order to understand the potential consequences of concentrated marijuana or THC use, we will look a little more in depth at the endogenous cannabinoid system. The endogenous CB1 receptor was first identified in the 1990s, and later investigation localized CB1 receptors in the brain at presynaptic sites, primarily on GABAergic and to a lesser degree glutamatergic neurons. The CB1 receptor primarily binds to the G inhibitory class of G proteins, shutting down adenylyl cyclase and cAMP production. The result of cannabinoid receptor activation is a reduction in neurotransmitter release as calcium influx is prevented in the presynaptic neuron. Naturally, endogenous cannabinoids like 2-AG and anandamide are synthesized via hydrolysis of membrane phospholipids and are then able to quickly diffuse across the membrane and act at receptors preceding the synaptic terminal. This process of retrograde signaling is implicated in long-term depression of the synapse, in which glutamate activation can permanently downregulate neurotransmitter release via phosphorylation of presynaptic regulatory messengers. This process can occur at a single synapse as well as heterosynaptically. Endocannabinoids are released in an activity-dependent fashion and have been implicated in learning and memory, appetite, and analgesic effects in humans. An important consideration when comparing THC action to that of endogenous cannabinoids is their respective efficacy at the CB1 receptor. The endogenous cannabinoid anandamide is a low-efficacy partial agonist like THC, and thus THC action in the body likely models that of anandamide at cannabinoid receptors. 2-AG, however, is a full agonist at the CB1 receptor, and as a result, THC can have antagonizing effects under conditions of low receptor density when this molecule is involved. This model of action can be observed in patients receiving the CB1 receptor antagonist rimonabant, who lack an increased heart rate response to cannabis use yet still experience a moderately attenuated high as measured by surveys with questions corresponding to symptoms of cannabis intoxication. This method of action may also play a role in how synthetic cannabinoids confer additional adverse effects. The shift in potency of cannabis products with the introduction of marijuana wax has heightened concern over psychotic side effects of chronic marijuana use, with numerous studies associating cannabis use with an increased risk for developing schizophrenia and other schizophreniform disorders in later life. On average, severe cannabis users were found to have a fourfold greater risk for schizophrenia and other psychosis outcomes than non-users. Increased cannabis exposure correlated with increased risk for psychosis in all studies that were analyzed. Additional experimentation amongst patients with psychosis has also shown that those with continued cannabis use experienced increased relapse in psychosis and associated positive symptoms when compared to both discontinued users and non-users. A potential avenue by which THC may induce symptoms related to schizophrenia involves the natural dopaminergic system, specifically the D2 and D3 receptors in the nigrostriatal system and nucleus accumbens. Currently prescribed antipsychotic drugs are known to target and block D2 receptors in the brain, and recent genetics research has also associated the D2 receptor gene with schizophrenia in humans. In vivo chronic application of THC in mice, even at low levels, was shown to induce hypersensitivity of D2, D3 receptors in the ventral and dorsal striatum. D2 and D3 receptor availability was measured using fluorescent receptor antagonists, as shown here, with significant increases in receptor concentration noticeable after 3 weeks of treatment with THC. Most noticeable is the receptor upregulation in the caudate, putamen, and nucleus accumbens. The dopamine system itself may be modulated by glutamatergic inputs, which can be subject to long-term depression as described earlier. NMDA receptor dysfunction, as well as NMDA receptor antagonists like PCP and ketamine, have been implicated in psychological effects resembling the symptoms of schizophrenia. In this sense, NMDA hypofunction can make the dopamine system more sensitive, disinhibiting dopaminergic projections from the midbrain through reciprocal action of glutamate and dopamine. The effect of THC on the system is specifically important during adolescence, and chronic exposure to THC during this period of brain development results in permanent changes and enhanced self-administration of a synthetic CB1 agonist in mice that reach adulthood. These findings illustrate that passive early exposure to THC has lasting effects on the reward system in the brain, and analogous effects in humans could lead to increased cannabis use or adverse mental health. The increased synthetic CB1 agonist intake may signify a reduction in rewarding effects, owing to decreased neurotransmission of cells from the VTA to the nucleus accumbens. Irrespective of the biological actions of THC, marijuana concentrate production and use has other ramifications on society as a whole. Production of butane hash oil, especially since recreational legalization of marijuana, has been associated with a drastic rise in the number of emergency burn accidents and explosions related to this method of extraction. The marijuana concentrates available to date are also not heavily regulated, and as a result have been found to contain residual solvent molecules or concentrated pesticides left over from the flowering plant. In fact, isopentane was found in 29.8% of tested wax samples, and the pesticide paclobutrazol was found in 22.8%. Processing of marijuana concentrates will be an issue of increasing concern as the legality surrounding marijuana continues to change. Regulation of concentrate processing will likely be an aspect of future recreational marijuana legislation, and whether or not such substances remain legal and available will likely depend on the increasing body of scientific evidence concerning the actions of these more potent drugs.